Environmental pollution, particularly the elevation of carbon dioxide levels, significantly threatens the delicate equilibrium of our internal pH. This situation underscores the critical necessity of sustaining a balanced internal environment. Buffer systems emerge as fundamental mechanisms in the regulation of pH levels, providing immediate physiological responses. These systems are composed of weak acids paired with their respective salts, effectively contributing to the maintenance of biochemical stability. The human body exhibits a greater capacity to compensate for acidosis compared to alkalosis. This distinction highlights the imperative of understanding and supporting the fundamental valencies that govern our physiological functions. The bicarbonate buffer system holds significant importance due to its ability to replenish itself through external sources, thereby sustaining pH balance efficiently. This system effectively neutralizes excess acids present within the body. Similarly, the phosphate buffer system plays a vital role within the renal system, engaging in acid-base regulation through a process known as acidosis. It contributes crucially to the maintenance of pH levels throughout the body. Moreover, protein buffer systems, exemplified by hemoglobin, exhibit the capacity to act as both acids and bases, contingent upon their surrounding environment. These systems are essential in the regulation of acid-base balance in the circulatory system. The formation of bicarbonate is integral for the transport of carbon dioxide in the bloodstream, effectively converting it into less harmful entities. This biochemical process transpires in both bodily tissues and the lungs, thereby facilitating essential gas exchange. Changes in ventilation have a profound impact on acid-base balance. For instance, hypoventilation can result in heightened acidity, while hyperventilation may induce alkalinity. The respiratory system adeptly compensates for these fluctuating conditions through adjustments in breathing patterns. In contrast, the kidneys play an equally important role in the regulation of acid-base balance, yet their response time is notably slower when compared to the immediate adjustments made by the respiratory system. Their influence on the body's pH can take several hours to days to manifest. Acidogenesis serves as a mechanism that governs acid-base equilibrium by facilitating the excretion of surplus hydrogen protons, utilizing phosphate buffer systems for effective regulation. This process remains essential for the maintenance of homeostasis. Aldosterone, a hormone synthesized by the adrenal glands, is responsible for conserving sodium while simultaneously playing a critical role in acid-base balance. Its actions promote the reabsorption of bicarbonate and the secretion of hydrogen ions. Disruptions in aldosterone levels can precipitate electrolyte imbalances, thereby affecting acid-base status. These imbalances may lead to conditions such as hypernatremia, hyponatremia, acidosis, or alkalosis. The kidneys are crucial in acid-base regulation, activating mechanisms such as ammoniogenesis, particularly during respiratory disturbances. This function ensures the preservation of homeostasis despite variations in pH levels. Furthermore, the relationship between carbon dioxide levels and acidity elucidates how respiratory issues can precipitate acid-base imbalances. This correlation accentuates the compensatory role of the kidneys in such physiological events. Acidosis can be categorized into gas and non-gas types, with non-gas acidosis often associated with renal dysfunction. This classification underscores the significance of renal function in maintaining metabolic balance. The logarithmic nature of pH, along with its resistance to minor fluctuations in concentration, emphasizes the necessity of precise measurements when diagnosing acid-base disorders. The partial pressure of carbon dioxide, or PCO2, serves as a fundamental indicator of both gas transport functions and acid-base status, significantly impacting respiratory health. Standard bicarbonate levels are vital for accurately understanding true bicarbonate concentrations, which can vary significantly under differing physiological conditions. Bicarbonate ions represent a principal component of the anions measured in the blood, reflecting the body's overall acid-base balance. Their levels can serve as indicators of underlying health conditions. The buffer systems present in the blood, including phosphate and protein buffers, play a pivotal role in maintaining normal pH levels. The presence of hemoglobin additionally influences the buffering capacity. A discrepancy in excess bases within the body is indicated by the difference between measured and normal buffer levels, which should not exceed 2.5 millimoles per liter. Such deviations may signal conditions like metabolic alkalosis or acidosis. Gas-related disturbances arise as a result of alterations in carbon dioxide concentration, leading to conditions such as respiratory acidosis due to hypercapnia. Specific scenarios influencing this phenomenon include inadequate external respiration and prolonged exposure to elevated CO2 levels. Conversely, respiratory alkalosis results from diminished carbon dioxide levels, frequently associated with hyperventilation. A comprehensive understanding of the causes and implications of both conditions is essential for effective clinical management. Respiratory acidosis can manifest through various mechanisms, such as inhaling air in confined spaces where carbon dioxide accumulates, leading to detrimental health effects. Recognizing these specific situations is vital for preventing respiratory complications.